we are ready to move to something different that is real, that has made a big change in the history of COVID. And I think it is vaccination. So if you agree, we can move to the last session about immunization. Am I right? So
yes, thank you, Eva. We are about to move on to a video, and this time it's Stefano who is describing his experience of having vaccines for COVID-19. And this is someone who has a lower immune function. So listening to Stefano will be incredibly beneficial to us all. So we'll head over to the video of Stefano. Thank you.
Right from the start, I've been involved in patient advocacy in the field of rare lung diseases and lung transplantation. I had a mild cold that triggered a drop of my spirometric values of over 20% within a short time. A drop not due to an infection, but more to a form of rejection inflammation. I was obviously very frightened at the idea of having to go back to oxygen and all the consequences of rejection. Talking to the doctor who has been following for 10 years after transplantation, I learned that this very large number of vaccine doses may have stimulated my immune system too violently, causing this form of inflammation. I would like to make it clear that I believe in science and that I started to take an interest in the COVID vaccine when it was not yet available in Italy, trying to push the health authority so that both lung transplant patient and patient with serious lung diseases could have priority in taking the vaccine. And I'm also happy to say that these authorities actually understood our needs by including us among the categories with priority. I personally underwent 4 doses of vaccine plus 1 dose of a preventive monoclonal drug and managed to produce a fair number of antibodies. The majority of lung transplant recipients, however, due to the high doses of immunosuppressants taken, did not produce any or very low quantities. The doubt that assails our community is why insist on this route with a 5th, 6th dose if there is no result in terms of number of antibodies produced? Should we not look for alternative methods for this type of patient? I would just like to point out how difficult it is now for us lung transplant recipient to make the right choice that protect us from COVID-19, but at the same time preserves the gift that we received through the transplantation. We continue to trust in scientific research, hoping that a solution will soon be found for us too. Scientists should not only propose 5, 6, 7, 8 doses, but also another alternative, no? I think only for us, because for normal people with a normal immune system, no, it is good, no. We have to know what could be the side effects for us, not for all the community, because we are different than all the community. This is what we want, more transparencies on the possible side effect for us.
Okay, so we are now moving to the next speaker that I'm really glad is here today with us, and I'm here to introduce her. She's Professor Nita Simons, Emeritus Professor at Imperial College London in the UK, and I have to say I'm particularly glad because she has been the source of inspiration for many of us within the European Respiratory Society, and not only because of her scientific value, but particularly because of the human value that is an extraordinary, I would say, combination. So I think she's perfect to talk to us at this session today about vaccination. Thank you, Anita.
Well, thank you so much, Ava, for those really kind words. I'm so grateful. I'm really glad to be with everyone this morning. I've enjoyed the conference so far and all the questions, the brilliant questions. So it's a bit of a tall order, 10 minutes to get through current vaccines. And I promise I will return in the presentation to some of those important points that Stefano raised. I hope you can see the presentation and hear me fine.
Perfect.
Okay, I will carry on. Just to say that my background is as a respiratory physician, but I became involved in vaccine and COVID drug regulation through working as a member of the Emergency Task Force of the European Medicines Agency, and I also belong to an advisory group for the regulatory agency in the UK. So what I'm going to do is run through what's new in COVID vaccines, that's SARS-CoV-2 vaccines, and the moving target of the virus, talk about the effectiveness of the vaccine in chronic respiratory disease, including in immunosuppressed individuals like Stefano. We'll talk about vaccines and long COVID briefly, and then always with vaccines, we want to know how efficacious are they and how safe they are. If we have time, we could talk about associated developments, monoclonal antibodies and antivirals, or we could just talk about those in the question time. So I'm going to start with a poll, and that is not what you believe, but what proportion of the EU population believes that COVID-19 vaccines are important? Which of those numbers, 87, 77, 57%? And I base this on very recent information just published in the last few weeks from the EU Commission. So it's almost equal there. We can end the poll and share the results. And you can see, yes, almost equal, in fact, slightly more, believing it's 57%. So if we could move on from the poll now. Just stop that. And in fact, the answer, let me just see if I can move on, is the middle one, 77%. So it could be a lot better. It also could be worse. You can see I've highlighted on that graph there the beliefs that vaccines are safe, and at the bottom whether vaccines are effective. There is quite a bit of variation across Europe in terms of those beliefs, and actually less belief in Eastern Europe than Western Europe, but still a fair amount of variation. And it's sad to say that the overall belief in vaccines has fallen after the pandemic compared to pre-pandemic. And of course, no vaccine is going to work unless you receive it. So what vaccines do we have available? Well, basically they fall into two categories. They're either univalent, which means that they're working against one strain of the virus, or they're bivalent, which are the more recent ones, which are working against 2 strains of the virus. And the original vaccines, as I think we'll all know, are the Pfizer-BioNTech one, the Moderna vaccine. They're both mRNA vaccines. And then there's the AZ vaccine, which is an adenovirus vector vaccine, the J&J vaccine, also an adenovirus vector vaccine, and the Novavax, which is a kind of subunit of the spike protein vaccine. And then in the last few months or so, we've had available, as I say, these combined bivalent vaccines, which include the ability to create a response to the original strain of the virus, the Wuhan strain, and then the ability to generate an immune response to some of the Omicron variants, so either BA1 or BA4 and 5, so two components within them. And we have received them, and people have been receiving them as boosters. Then there are other vaccines still in the regulatory cycle, in the process of being approved. Hipra vaccine, the Valneva vaccine, which is a whole vaccine, but there's a sort of protein within it to stimulate the response. And of course, we know there are Chinese and Russian vaccines like the Sinovac and the Sputnik vaccine, not used in Europe. The research that's going on at the moment are looking at different targets within the virus of the vaccines, trying to understand the immunology of measuring the response, because now we're not doing wide trials amongst large groups of patients with the boosters. We're comparing the immune response to previous immune responses. There's the appropriate timing of the doses. So we now know that leaving a longer gap between the initial 2 vaccines can be more beneficial. And I think relevant to the question that Stefano raised about vaccine delivery is there is a lot of work on intranasal delivery of the vaccine. So that just like there is intranasal delivery of the influenza vaccine in children, that's it. Delivered directly into the nose, that may be helpful because that is, after all, where the virus gains access to the body, and generating a mucosal immunity might be more effective than delivering it, say, into the arm. That's yet to be proven. And then, which would be really helpful practically, combination vaccines, like having together influenza and a COVID vaccine in one dose. But we have to remember that we've got this moving target here because the virus continues to mutate. And these are data from ECDC, European Centre for Disease Prevention and Control, in the last week, showing at the moment these different countries across Europe and the different sorts of variants. So as we heard from Tobias, at the moment, it's predominantly the Omicron variant as opposed to the original strain, then the Alpha and Delta variants. And the dark green here in the different countries is the BA5 variant. So you can see that's really very dominant in Europe at the moment. The purple here is where it's difficult to differentiate between the BA4 and 5 strains of Omicron, and the lime color is BQ1, which seems to be creeping up in some countries, but certainly is not dominant at the moment. And there are a few other variants of interest and concern that we're watching for, like this BA2.75. I don't have time to dwell on it, but the ECDC gives interesting information across all countries on the case numbers. This happens to be UK with a spike. You can see here in the summer, in July, another spike last month in October, and the predictions for December, relatively low case numbers. And this is almost identical, say, in the Netherlands, in Spain, similar spike, not the later spike here. So this is where we are on trying to keep track of the virus. So when we talk about vaccines and how they work, there's 2 words that are used. It's always a bit confusing, but it's vaccine efficacy, and that's the number you see coming out of the trial. So they say the Pfizer vaccine is 93% has 93% efficacy, or the AstraZeneca virus vaccine has a just under 80% efficacy. But in the real world, we use the term efficiency because that's the way the vaccine works in broader groups of people and people that are not necessarily included in the trials. Also, when we're looking at the prevalence, so the number of people affected in the real world, there are a whole number of factors that are taken into account, such as the vaccination status of others. We talked earlier about the impact of shielding and lockdowns, the fact that the variant of the virus has evolved over time, so trying to escape control by the vaccine. I just want to add a quick word on shielding because I know so many people have sacrificed so much to shield. The more recent studies of the population of a whole have shown that it's not that beneficial. But that is because shielding is always imperfect. Perfect shielding, if you do shielding perfectly, works. There's no reason why it shouldn't. But when we look to the population, to the real world, there are many factors that make it very difficult to shield. For example, intergenerational families with grandparents, parents, and children, very difficult to shield in those situations. We know that there are key risk factors, older people being affected more, genetic predispositions, occupational, so people, bus drivers, paramedics, people working in the accident and emergency department, and the strong correlation with social deprivation and ethnicity. So we heard earlier too about prevalence in different disease conditions, so I won't go into that, but a really important issue we have with the vaccine and with coronaviruses. So COVID is a coronavirus, and it's really difficult to produce a vaccine that's very long lasting against coronaviruses, unlike, for example, a tetanus vaccination or a pneumococcal vaccination, which is going to last longer. And you don't have to understand these graphs. You can almost look at them through your eyes half closed, but you can see this is the vaccine effectiveness over a period of time. And this period of time is over 7 months. The top three graphs are in people of all ages, and the bottom graph is in older individuals, those over 70. And in essence, you can see a falling off of effect over time, but importantly, a real takeaway is the effect is much better maintained, and actually maintained pretty well with some vaccines, against severe disease or hospitalization, as opposed to any infection. So asymptomatic infection, symptomatic infection, the protection wanes more quickly. And that is the issue we have, is that it's not so effective at people getting infection, but it does stop you becoming more seriously ill. Now, what about the effects of the vaccine in people who have immune conditions? So they may have a primary immune condition, or they may have a condition, interstitial lung disease, some other condition where you are on immunosuppression to reduce the effects of that condition. And this plot is showing you along the central line here, this is showing whether, if your condition, you have the same ability to seroconvert, that is to get protection from the vaccine, as someone without that condition. You can see throughout a range of those, all different sorts of conditions with immunosuppression, that reduces your ability to mount a response. And that ability to mount a response is even more significantly affected in the transplant recipients, although interestingly was not as significantly affected in people with HIV. Now, we talked about boosters, and Stefan said the booster doesn't affect. In fact, the evidence is that in many people, and this is solid organ transplants, there is evidence that the booster, so this is after 2 doses, your primary series of vaccine, and this is after the 3rd dose, so this is the first booster, in many people there is a worthwhile response. It does vary, it's obviously better in some than others, but that is the logic, the argument to have boosters, that it does have an effect. So boosters help by and large. I don't want to go in detail here, but we tend to measure antibodies and seroconversion, but actually this study is in heart-lung transplant patients. So this is a response. So looking at antibodies in people who are heavily immunocompromised because of heart-lung transplant. This is the response in healthcare workers with normal immunity. You can see a much reduced response, but this is T-cell response as well. And actually, T-cell response may be important for your memory to fight the next time you're exposed to the virus. You can see here that although the T-cell was reduced compared to the response was reduced compared to the healthcare workers, the different vaccines didn't make too much of a response, and the response was somewhat better. So we need to remember that it's not all about antibodies. Just briefly, so I don't run over time too much, vaccines and long COVID. Well, Joanne mentioned a little bit about this, but COVID vaccine almost halves the odds of developing long COVID, so it's well worth having the vaccine. I know many people developed long COVID before the vaccine came along, but even in that situation, there is some evidence that having a booster or subsequently having a vaccine series can produce some improvement, and more people showed improvement than felt they deteriorated. So there's good reason to have the vaccine and to have the boosters. I'm just going to quickly mention a really important point about the safety of vaccines. Of course, we know a lot about side effects from the trials, the large trials that are carried out. And then for every vaccine, there is a comprehensive program of post-marketing. So once it's available to everyone, of surveillance, and there are yellow card systems. There's a huge vigilance monitoring system in Europe. AI is used to sieve out and focus on reports that are coming up more frequently. And from the European Medicines Agency, for example, there are monthly adverse effects reports. We have come across several important effects of vaccine. So associated with the adenovirus vaccines, the AZ vaccine, the J&J vaccine, there is an important adverse effect, this vaccine-induced thrombosis and thrombocytopenia. So people end up with blood clots. It is rare, so you're very unfortunate if you get it, but that's why there is less AZ vaccine used. With the mRNA vaccines, myocarditis and pericarditis in young people, teenagers, and people in their 20s, more in men than women. Most of this recovers with very little intervention required, and then some other conditions that are seen more often in those who are vaccinated, but at very low levels. And one always has to bear in mind relative risk, because clotting, as we've heard, myocarditis, pericarditis occurs with COVID. And so you're always weighing up the risk of COVID versus the risk of side effects with the vaccine. And then to go particularly to Stefano's point about can having the vaccine actually cause rejection in those who have had transplants. Well, this is a very recent study. It's a review of all the cases in the literature of people who have developed evidence of rejection of transplant, either after COVID vaccination or following COVID infection itself. And bearing in mind, there have been many millions of vaccine doses in the literature at any rate. So published results, you can see there are cases after the vaccination, but there are also cases after COVID-19 infection. The majority of cases are actually for corneal grafts, as opposed to next being renal transplant, and then relatively low at heart-lung transplant. And most of these, there was evidence of increased inflammation, increased evidence of rejection, but that was treatable, and the organ itself wasn't rejected. This won't cover all the cases because not all the cases are reported, but I think relatively reassuring that the risk is low. So that was all I had to say. I have an extra slide that I can come back to if you wish on monoclonal antibodies and antivirals, but I'll only do that if that's what the chairs wish. Thank you very much indeed.
Many thanks, Anita. Again, incredibly interesting and informative. And it's good to see the variety of vaccines that are available and that they might help with long COVID. And that's certainly very reassuring. So thank you for that.
But I think I would start. There is a lot of discussion. We could be a bit out of time, but I think we like discussing here today. I will invite the speakers, please, to stay with us. And I think we'd like to ask you two main questions that are not only for you and Anita, but I think also for people in the audience. So if we did not achieve the goal of complete vaccination, despite the great evidence in terms of efficacy and change in the natural history of the disease, what failed first? Second, what do you think we can do better in the future? This is a question for everybody. And I think, most people might want to say something, have, personal experience and scientific background, a combination of things that might help in this sense. You want to start, Apostle? What failed in your opinion?
I think that there was in our patient, the majority, they were very positive, but they have concern that the vaccine goes too quickly. And I think it failed the success. As everybody knowing ourselves in research, it was a miracle that it did that. But the patients should know that as long as physicians, also researchers, we stop doing everything else. We stop doing anything else. So that's why they come up so quickly, because we go all the steps in parallel together and very quickly, work night and day. But we didn't make it more easy. So we were serious. I think that was the one. And then I think now that we know what's happening, and the next time we should discuss with the patient. We have this, I think this is very important, to listen each patient, what she or he thinks about the vaccination, and take this very seriously and take the possibility to discuss with them and persuade them. Very simple, but very effective. Thank you.
Anita, what failed in your opinion?
Yeah, I think several things. Firstly, on the swiftness of development, I think probably the regulators weren't good enough at explaining that a whole different process was used, this rolling review. So instead of the approval happening in stages of the sort of early studies, the animal studies, and then the large human trials, there was a lot of understanding the information as we went along. So it was a very dynamic process, and that's why it was so much quicker than the previous development of vaccinations. Also, a lot of the companies invested a lot of money in already developing the vaccines themselves as the process was going on, so they took on the risk. Another issue, I think, is that we weren't clear enough. I think, as Apostolos said, the vaccines were going to be a total magical solution, and what we've ended up with vaccines that are very effective against severe disease and ICU admission, but don't prevent transmission. And I think that was something that wasn't understood. It wasn't anyone's fault, but it wasn't understood at the beginning. And then thirdly, I think we have to say there have been very significant misinformation and disinformation campaigns against the vaccine. Thank you.
Thank you. So what can we do better? Apostolos.
Well, I think that we can, first to discuss with our patient, that we, as we said, but also I think each of us, we have also responsibility beyond our own group. So we should go out, as Anita said, and face this misinformation, to be active. And this is, I think this is a huge work for ELF, and they already do that, for the patient and from us, ERS and the other societies. But I think we should be very proactive about this stupid information.
Yeah. Foggy news, right? What about Anita? What do you have in mind to improve vaccination and people trust in this technology and this preventive measure?
I do think it's individualizing, and actually what the conversations that you have to start from the basis of people are saying they're worried about something and they're worried that it's going to affect their chance of getting pregnant in future, or they're worried about having it for their children. You have to understand that they're coming from a point of actually caring very greatly about themselves or their child and so on, and acknowledge that first, and then you confront that particular concern. I think from the whole vaccine confidence project that, you know, Apostolos alluded to, and that is there is a group of people whose beliefs are so extreme that investing time discussing with them probably gets you nowhere. And so you have to focus on the vaccine hesitancy as opposed to the people with an extreme belief system. And it's very difficult to know what to do about that. And it's actually undermining the community's belief in other vaccinations, such as childhood vaccinations. So it's a really, really important problem. I'm not sure if I've answered it at all.
And Anita, could I just ask a question that's come up in the chat? How likely is it that we would get a vaccine that would deal with the variants, all of the variants? So rather than getting repeated booster vaccines, what's the chance of a vaccine that is likely to have a wider coverage?
There is a chance that that will happen, but it's we're only just trying to get there with the influenza vaccine. We still accept that we take that every year and that the virus mutates, and we have to adjust to that. So I think there is the possibility of trying to deal with rather than particular aspects of the spike protein where it attaches and enters the cell. Also, I think the intranasal delivery, so trying to stimulate this mucosal immunity might be more successful too. But we're a little way from that at the moment, Helen.
Thank you for that. Just having a look at the chat to see if there are any other.
I've seen another question about the role of antibodies, monoclonal antibodies, and their role in preventing the infection in immunodepressed patients. Can you tell us a bit more about Evusheld and other opportunities for patients with this condition?
Yes, that they do have an important role in people who are immunosuppressed, and monoclonal antibodies can be used to treat individuals who've got COVID. They can be used in a preventative sense as well, either just to give it anyway, or if you're a contact, so someone in your family has it. Again, the monoclonal antibody has to reflect the predominant variant, and we've been through some of the monoclonal antibodies to the original strain and to earlier strains do not work well against the current sort of BA5. So Trivimab is one that is being used. That's what's being used in the UK. There is an issue with Evusheld, which is a combination of monoclonal antibodies, two monoclonal antibodies, and it's not approved for use, full use in the UK and some other countries, although paradoxically, and I think you know, very difficult ethically, in some countries is available privately. The reason it's not fully approved is that the efficacy of it against the current strains is not brilliant, but you have to have the right monoclonal antibody in essence.
So in this sense, I think update of research and the monoclonal antibodies available will be determinant, because as the strain the virus is changing with new strains, possibly we will need update not only vaccines but also this kind of therapies. The other option we have is Paxlovid, that is for treatment in the early phase of the infection. How important is that treatment in your opinion at the present time of the pandemic?
So the antivirals are interesting. So these are specifically for high-risk individuals, and the it does work. There are some problems with it. You can get rebound. So initially it works and then the COVID seems to come back again. There's also quite a lot of interactions with different medications. So I think it may be, and it should be available in high risk individuals. And there are one or two other antivirals as well. Molnupiravir is one. Remdesivir has a sort of borderline efficacy as well. The issue with antivirals is they have to be used early. You must use them early in the course of the disease.
Yeah, that is the key point, because that's been proved through a specific clinical trial where it was, demonstrated to be useful in early phase of the disease. That does not make sense, or we cannot give any feedback on its efficacy in later phase of the disease. So it's going to be crucial to know how to use it in the future. But as it has been stated many times, what is key is communication, interaction, collaboration, and research. We need to do more and more research, because we have not finished. COVID is still there, we don't know how long it will be concerned, depending on how it evolves, right? So I think collaboration is going to be key. I think we have a lot of questions, but as we run out of time, I think I would like to ask ELF office to show a key slide to show you how we can keep in touch to conclude, and also to conclude this session, because I can imagine, I hope you have enjoyed this session. I hope a number of questions have been answered, but I'm pretty sure there is still a number of questions that have not been answered. So I really would like to invite you to keep in touch with us through a link we can share with you. Can we? I'm not sure you are seeing the right slide.
Yes, the slide's up with all of the links.
Good. You can keep in touch with us through the links on the slide. So sign up to the ELF newsletter, get involved by emailing us, engage through social media, and find out more about the Dragon project. It is a specific European project dedicated to COVID, to its management, and we can find more detailed information on our Dragon website. So thank you very much. The engagement has been incredible. I've seen a lot of positive feedbacks during the sessions. So really, I'm really grateful to the speakers and to people in the audience moving up with so nice questions. So keep in touch, and please send us your questions if you still have. We will try to answer as soon as possible. And this interaction can only be productive and helpful for everybody. So thank you very much.
Yes, I just want to do a final thank you. And I want to thank the speakers especially for your insight, for your engagement, and for the information that you've been providing to us today. It's been invaluable. I'd also like to say a word of thanks to the COVID-19 Patient Advisory Group, the Dragon team, the ELF team that are behind the scenes doing all this work for us. They are incredible, and they really have helped to promote the event. And I thank you if you've promoted it and continue to do so. All of the talks will have been recorded, which again is incredibly helpful. So if you're posting on social media, make reference to that, because I've no doubt a lot can be gathered from the information given by each of our experts today. Just to say, a short form will be sent to you, a feedback form, if you've registered, just to give us some feedback on the event today, and also to help keep you involved in future events. So thank you very much to everyone for all of the efforts today, and I hope you enjoy the rest of your weekend. Thank you very much. Goodbye.
Thank you all. Have a good day. Bye-bye.